Hi everyone, it's Mr. Sinti, and today I'd like to talk to you about the process of transcription. Yeah, transcription is one of the key processes that a cell needs to do in order to create RNA. That's right, the creation of RNA is important because it acts as an intermediate in the process of protein synthesis. And so transcription involves this discussion of turning DNA into RNA, and then RNA is going to be involved in the translation process, converting the nitrogen-based sequence into a polypeptide sequence. And so this conversation that I want to have today in this particular discussion is just on transcription and how it works. And I'm going to take sort of a look at simple transcription in prokaryotes or bacteria, and then look at a little bit more complex situation when it comes to eukaryotic cells, especially how they can regulate transcription. It's pretty cool. So right out of the gate, I just want to say that transcription is the process of synthesizing RNA. And so what we're doing is, this is a polymerization. Basically, we're making the polynucleotide ribonucleic acid. And we're making all kinds of RNA. We're making messenger RNA, transfer RNA, ribosomal RNA, small nuclear RNA, interference RNA. So all RNAs are produced in the same process. And they're transcribed from a template which is on DNA. And the overall operation of this is kind of simple. It involves a very important enzyme called RNA polymerase, appropriately called. And so what does this do? It attaches to DNA, causes the DNA to open up. In other words, the double helix unwinds, and then what it's capable of doing is bringing in new RNA nucleotides. So those are phosphate sugar base, bringing in those 4 RNA nucleotides and extending a new piece of RNA from the template strand in a growing 3' direction, and eventually we're going to reach a termination sequence on DNA, and the RNA is completed. And so you're like, well, okay, that sounds pretty straightforward. Let me show you this. So here's a picture of DNA, and as you may be aware, it's tightly coiled around these interesting proteins found in the cell, in the nucleus, called histone proteins. And so this sort of beads on a string is called the nucleosome. And so here comes the enzyme RNA polymerase. And so what it's going to do is it's going to bind to a particular area. And this region of DNA where it binds is going to be like a big part of our conversation. The part of the DNA for which it binds or recognizes is called the promoter sequence. And so this is where it binds, and then it starts to transcribe. And this red structure that's coming off here, it's not showing any detail, is RNA. And so what it's doing, it's bringing in new nucleotides from the nucleoplasm, and it's producing RNA. So it's able to bind and extend or elongate, and there's the RNA. And so here's a little detail of what's happening. Here's RNA polymerase. These red structures are RNA nucleotides, and the enzyme is bringing them in, and it's complementary to the template of DNA. And so as you can see here, it's attaching them in a growing 3' direction. It has to be in the 3'. And so here, if there's a guanine here, then the cytosine will come in. So I just wanted to show you that particular overview before we get into any more detail. And so I hope you enjoyed that. And so what we're talking about is the making of RNA, and the enzyme involved right here, this big large guy, is RNA polymerase. And so like DNA polymerase, RNA polymerase adds new nucleotides, but it's choosing RNA nucleotides, and it extends them in a growing 3' direction. In other words, it attaches it to the hydroxyl group on the 3rd carbon. And so I wanted to point this out. It's fairly significant that the genes on DNA are on the 3'/5' strand, meaning that when you create messenger RNA, you're doing it in an opposite direction. So in other words, here's the DNA double helix. So when this opens up, what you're doing is this right over here is this particular sequence. Let me come back to that. This sequence of DNA right here is where the genes are located. Okay, because this is what's being transcribed. So it's A, U, G, C, C, A. And that's significant because when you consider it, this is now the 5' end of messenger RNA, and the code is read in groups of 3. So this is a codon, and a codon, and a codon, and a codon. And then if you had your decoder with you, you'd know that AUG on messenger RNA is the amino acid methionine, and then proline, etc. And that's how this is called translation. So what I want to focus our attention in this video is how do we produce RNA? That's what I want to focus in on. And so what I want to say is, and I've mentioned this before, is that there's particular sequences along the DNA, like this. If here's DNA, there's a particular sequence, and I'll just highlight it right here in green. So a segment of DNA that is called the promoter. Now the promoter is usually upstream of the gene, and what that means is that it's just a little bit before the transcription unit, or the gene. So here's the gene for a particular polypeptide. So this is the gene located here. Here's the promoter. Here's the gene. And then usually there's some kind of termination sequence that on DNA, termination sequence, terminator sequence, that marks the end of transcription. Now, this is a little bit more understood in prokaryotes, truth be known. There's a particular sequence that usually ends the transcription in bacteria, but sometimes the termination sequence in eukaryotic cells are a little bit less known or unclear. It's sort of like where the RNA polymerase stops, no one knows. Kind of an interesting thing. And so, as it turns out, again, most of our understanding of these kinds of molecular issues have come from bacteria. And so we're more confident about speaking of prokaryotes. So bacteria have a single type of RNA polymerase. They have one, and it does all of the RNA that the bacteria need. So it's a single RNA polymerase. It's kind of nice. However, for our cells, since we're eukaryote, we have 3 different RNA polymerases. Now, I know that may seem detailed to you, and it's like, well, a lot of things are detailed, so you can take anything with a grain of sand, but I'm not sure if that word, if that analogy works. But at any rate, there's an RNA polymerase 1, and there's an RNA polymerase 1 for rRNA, so that's ribosomal RNA. And as it turns out, that there's a lot of rRNA that's produced in cells. And so there's a lot of RNA polymerase 1. You need to produce each cell needs to constantly be producing rRNA because you need many ribosomes in order to make the proteins. RNA polymerase 2 is for messenger RNA, and this carries the genetic code. And then RNA polymerase 3 is for transfer RNA. Transfer RNA, as you may know, transfers the peptides to the ribosome in order to create the peptide bond during translation, or the making of a polypeptide. But what's of note is that especially in eukaryotic cells, the genes can be rather long, and as it turns out, RNA polymerase is somewhat slow as it's moving along because it has to carry that RNA transcript as it goes. And so it would be a little bit too slow to create all of the RNA necessary. And so I find this particular fact to be kind of interesting, though obscure, is that on DNA, there's many genes of the same kind. So in other words, these are repeated genes that are often associated with one another, and they're known as tandem repeats. There's many copies of rRNA genes in eukaryotic cells in order to produce the copious amounts of rRNA, because RNA polymerase is a little bit slow and you really need to have a lot of ribosomes. Kind of interesting. So again, here's our picture of the DNA, and that there's some specific regions that I want to highlight. There's this promoter region, which is where RNA polymerase recognizes and binds. Now, it doesn't do that alone in a eukaryotic cell, just pointing that out, but it binds to the promoter, and then it starts transcribing. So this is known as initiation, where it attaches. And then it starts elongating at the starting point of the gene, and the pink is the messenger RNA, or the RNA in general, the transcript. And then RNA polymerase will move along, elongating, making the RNA until it reaches this termination or terminator sequence. And so that's like the step 3. So it's initiation, elongation, and termination, and then you have this RNA transcript. And again, in a bacteria, the gene is made up of just all good DNA, meaning exons, that all the sequences are then going to be an RNA, and then those are expressed into the protein. In a eukaryotic cell, there's a separate video on this, but the gene is interrupted by regions of noncoding called introns, and those need to be processed and removed and spliced out. Again, separate video on that. And so I just want to talk about transcription today. And so the emphasis is on this promoter. And so here's a typical promoter in a prokaryote. And a lot is understood about this. And so I mentioned there's this start point where the gene is located, where I'm emphasizing right here is a start point. But what's upstream of the start of the gene is this promoter sequence. Now the promoter sequence, if you go in reverse order, so if this is 0 or 1, this would be -1, -2, -3, -4 if you go upstream. So this -10 sequence is sometimes referred as, because it's rich with thymine, adenine, thymine, adenine, thymine, it's sometimes called the Tata, because it sounds like that, Tata box. And so this is a place where RNA polymerase actually attaches and like hunkers down and nestles into this region right in here. And then there's a sequence of about 15 or so bases, and then there's, at this -35 sequence, this is another critical area where there's another protein associated with RNA polymerase called the sigma factor. And this area is slightly variable depending on whatever the gene is in a bacteria. And so the sigma factor helps to regulate whether or not the RNA polymerase is actually attaching or not attaching at that particular point. And so with regard to the 10-10 sequence, it's highly conserved. And so this sequence is usually the same in every promoter in a bacteria, because it's, like I was saying, it's where RNA polymerase actually attaches. But this -35 is kind of variable, and there's a family of regulatory proteins called sigma factors that are actually help RNA polymerase know whether or not it's to be transcribed or not to be transcribed. So they help to regulate transcription by allowing it to bind or not bind in this particular sequence upstream in the promoter. And so just a little background on that. So these bacterial RNA polymerases were first discovered by Professor Chamberlain at the University of California, Berkeley. And what's known about them is that they're fairly complex, but more simple than a eukaryotic RNA polymerase. So there's these 4 basic core subunits that are called alpha, beta, beta prime. And the 4th one is sort of these sigma factors that produce the variability. And so these sigma proteins will help RNA polymerase recognize the variable sequence at the -35 region of the promoter. And so simple cartoon of that. So here's the sigma factors and here's RNA polymerase, and together they associate, if they're both present, whether or not the gene will be transcribed or not. Okay. Now in eukaryotes, the RNA polymerase is gigantic. It's huge. It's so massive. It's made up of approximately 13 different subunits, and so it's a massive enzyme. And so it's able to then, not only that, it interacts with hundreds of other proteins at the promoter. So the promoter is this gathering place, sort of like a big metro station where it's the RNA polymerase is arriving, and there's all of these other hundreds of proteins all arriving at the promoter. And it's pretty interesting. And all of that has to do with whether or not the gene is to be transcribed or not. It's the way in which cells regulate the making of a protein, whether or not the RNA will be transcribed or not. And so there's a large number of what are called in general control elements. So these are segments of DNA that are non-coding, and when I say that, meaning like they don't code particularly for the gene, but they help to regulate transcription by allowing what are called protein transcription factors to bind and therefore allow the RNA polymerase to be attached to the DNA. And so let's get into that conversation. So in eukaryotic cells, there's the promoter, and again, there's this Tata box right over here. And so there's a whole group of proteins called transcription factors that are produced, and these transcription factors begin to attach in the region of the promoter. And then once these transcription factors have attached, they then facilitate the arrival, or they mediate, if you will, they mediate the arrival of RNA polymerase. And so RNA polymerase won't be able to attach unless these transcription factors are present. And it's showing RNA polymerase 2 here, which means that the RNA that's being produced is going to be messenger RNA. And so again, all of these proteins, and there's even more, create what's known as the transcription initiation complex. Okay, so all of this is happening during initiation, the first phase of the 3 phases of transcription. So what happens is you get these multiple transcription factors binding to DNA before DNA polymerase can actually do so. And so once they attach, then the RNA polymerase is capable of attaching to the promoter. Now, what was also discovered, I'm going to point this out here, is that, okay, here's the gene, here's the promoter sequence, and here's this little circle represents RNA polymerase. So what's interesting is, okay, everything's fine. But as it turns out, when scientists were experimenting with cells and trying to figure this out, what's interesting is really far upstream from the promoter, and the promoter again is where the RNA polymerase is attaching, scientists were able to delete segments of DNA really far away from the promoter. And as it turns out, when you do that, you get a very low expression of protein. And so that suggests that there's something far away from the promoter and the gene itself that has something to do with transcription, the making of RNA. And so as it turns out, this particular region was termed an enhancer segment of DNA because it enhances the production of protein. So it's called an enhancer. And as it turns out, if you delete sequences that are fairly close by, you can still get good production. So these enhancer sequences are pretty far away. And it's like, well, what are they doing? Well, as it turns out, if you were to delete the promoter sequence, you don't get very good protein production. And that could make sense because even though this region is intact, you still need RNA polymerase, the ability to bind in this area. And then some curious things were done in these enhancer regions is that you can cut them out and then put them in different orientations, and they're still able to somehow promote or enhance, I should use the word enhance protein synthesis. And not only are they found upstream, but sometimes they can be found pretty far downstream of a gene as well. And so this is kind of weird in terms of the discovery of these things called enhancers. And so let's talk about that. So in addition to RNA polymerase being dependent on transcription factors, there's these control elements called enhancers that may be thousands of nucleotides away from the promoter, even usually upstream, but sometimes downstream from the gene. And so what I'm talking about here is here's the promoter and here's the gene. And you're like, how can something so far away have anything to do with the promoter and the gene? As it turns out that these enhancer sequence, it was determined that there's also some transcription factors, these small proteins called activators, which could attach to these enhancer regions. And when they attach to the enhancer regions, you ready for this? That once these initial transcription factors attach, then the activators will attach to the transcription factors which would then cause the whole thing to take place. In other words, RNA polymerase of attaching or not attaching. And it's like the whole DNA can loop over like this. And it's like, okay, so summarize this. For transcription to take place, you need transcription factors, which are these guys, and you also need activators, which are attached to enhancer regions of DNA, which are pretty far away. But it's not just this. There's other proteins involved as well. There's things called co-activators. And then, watch this, then there's a group of, okay, here's the activators. Then there's a group of proteins called mediator proteins, which further, I mean, you can call the whole thing transcription factors, but they're mediator proteins that actually also help the RNA polymerase hunker down to the promoter. And it's these mediator or co-activators that attach the activator. And then I like this one. There's a particular protein that was discovered fairly recently that actually sits into this bend right here, and it's called DNA bending protein. So all of this, I mentioned there was quite a few proteins at the start of this video, so you shouldn't be too surprised. So all of this is allowing RNA polymerase to bind. But are you ready for this? There's also a protein called a repressor that can bind to the enhancer region, which would prevent activators. And there's also a repressor protein that can bond over here just adjacent to the promoter, which will prevent these transcription factors from binding as well. So there's transcription, there's regulator proteins that are involved in the process. Now, let me just drive right through this. Now, once RNA polymerase attaches, that's the challenge, the initiation. Elongation is actually pretty simple. Once RNA polymerase starts cruising, it starts adding new nucleotides in a growing 3' direction at about 10 to 20 bases at a time as it moves along. And so the enzyme can add nucleotides at 60 base pairs a second as it moves along. It's slower than DNA polymerase, again, because it's sort of carrying the rest of the molecule with it. And so behind this, the point, RNA synthesis, the double helix reforms, and the RNA peels off. Now, here is a close-up of that process, the new nucleotides adding, and here's RNA polymerase. Now, here's another picture of RNA polymerase attaching to DNA. Now, as it turns out, many RNA polymerases can attach to the gene at the same time. And then, how does it ever end? Okay, so, well, the length of the new strand reflects how long elongation has been taking place. So if it's a big piece of RNA coming out, it's been going on for a long period of time. And so it's longer, it's as long as it's necessary. If the protein's long, then the RNA transcript is long as well. And so where it stops, in other words, termination is, I mentioned this before, it's known in prokaryotes. It reaches, and I'll go over this first, it reaches something called a terminator sequence in DNA. And not only does it reach that, and which ends termination, there's another protein involved called Rho, which helps the RNA polymerase to become derailed from the DNA. But I find this to be kind of interesting. I hope you enjoy this. When the RNA polymerase reads on the gene, the termination sequence, what it does is it produces this sequence of messenger RNA. And it's like, well, what is this? Well, as it turns out, do you notice that this sequence right over here, and this is messenger RNA, is palindromic to this one? In other words, it's complementary to it. So when RNA is coming off like this and coming off over here, what will happen is it'll form this loop. Sometimes it's called a hairpin loop. And what will happen is, because the GC, AU, CG, CG, like that, and there's like a little space in between here, because of the fact that they're complementary this way, they'll form hydrogen bonds. In other words, there'll be a double helix region. And that whole structure, along with the Rho protein, causes the RNA polymerase to terminate. And so that's how it finishes, because of this particular sequence. Now, in a eukaryote, maybe someone knows, but I don't. Apparently, the sequence, there is a termination sequence, but where it really stops, sometimes RNA polymerase goes beyond it, and it's not quite clear on how that's happening. And so let me show you here a couple of animations that might clarify some things. And so let's see this one first. And what I want to mention about this is here's the gene right here, the coding region, and this blue area is the promoter. So let's take a look at this. And so what you're going to see first is for RNA polymerase to actually attach, because that's what it does to the promoter region, it has to initiate with a group of proteins called transcription factors. And so there's this one particular transcription factor called the TATA binding protein, and then these are alpha beta transcription factors. And so there's a bunch of them that assemble in this particular region of the promoter, and they're thought to help attach and sort of hold with this little arm the RNA polymerase on the molecule. And so once these attach, I mentioned to you before there's other proteins that are associated in this transcription initiation complex. These things are called co-activators, and here are the activators. Now remember, these activators, they sort of look like a lobster claw. They bind to these coding sequences called enhancers, and it's the enhancer that allows the activator to attach to this, and when there's enough activation, that will allow transcription to take place. That is, unless there's a repressor protein. Now, a repressor protein can actually bind to a region in the enhancer called the silencer gene, and that'll prevent the activator from actually attaching. So no activator, no transcription. And there's also the repressor protein will bond to a region sometimes very close to the promoter as well. Let's take a look at this particular video, and I'm going to let this one actually play out, and I'm going to put some sound behind it. This one's particularly good.
The central dogma of molecular biology: DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism.
So that yellow molecule is the
RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.
Until it reaches the termination sequence. So that's kind of cool. Now, it may not just be messenger RNA. It could be any of the RNAs that are produced. And so that's pretty interesting. And so, and then finally there's this.
In eukaryotes, DNA transcription begins when general transcription factors bind to the promoter region of the gene. Those transcription factors facilitate the binding of the RNA polymerase enzyme to the start position of the gene. RNA polymerase needs to be activated by a type of transcription factor called activator. Some activators recognize and bind to the regulatory region of the DNA near the promoter region. Other activators bind to a distant region of the chromosome called the enhancer region. Activators bound to the enhancer region work with mediator molecules to initiate transcription. Another type of transcription factor, called repressor, inhibits the activation of transcription. Repressors can act in different ways. Some repressors bind to the binding site of activators and prevent activators from binding to DNA.
Sort of silencing them.
Some interfere with the molecular interaction between the activators and the RNA polymerase. Some activators and repressors act by affecting chromatin structure. These repressors can alter the chromatin structure to make it coil up tightly. Coiled up genes are not available for transcription. Conversely, activators can uncoil a coiled section of DNA and make genes in those regions available for transcription. The interactions of all these factors determine the actual rate of DNA transcription.
So in general, I hope this wasn't too too tricky. I mean, it's not the easiest thing to discuss or understand. But safely, let's say this: that what we're talking about is an amazing process in which eukaryotic cells can regulate transcription by allowing RNA polymerase to attach or not attach at a promoter, and therefore you can regulate RNA production, and if you regulate RNA production, you then regulate protein production, and then cells can produce proteins when they need to and not when they don't. And so it comes down to a simple concept, but the details are rather tricky. So hopefully this was illuminating, and thanks for watching.